So this video will cover all of the organic molecules in basically chapter 3 of the Mastering Biology. And alright, so we'll start off with just what is an organic compound. An organic compound is basically any compound that contains both carbon and hydrogen bonded together with covalent bonds. So the major organic compounds that we'll discuss are carbohydrates, lipids, and proteins. And nucleic acids. And so we're going to start off with carbohydrates. So carbohydrates consist of carbon, hydrogen, and oxygen, CHO. And hydrogen and oxygen are usually present in a 2-to-1 ratio. So, basic unit of CHO is a simple sugar or monosaccharide. Monosaccharide are, like we said, simple sugars, but the ones that we will discuss come in pentose sugars, which is a monosaccharide containing 5 carbons, or a hexose sugar containing 6 carbons. Look at the prefixes if you need help. Remember, pent means 5. Think of the pentagon, and hexo, or just hex. Think of 6. So here we have a model of glucose, and as you can see, it looks like to be a hexosaccharide, or a hexose, and this is an important, very important actually, diagram, so try to remember that if you can. So then we have hydration synthesis. So I'm going to use this example here to discuss hydration synthesis followed by hydrolysis. So hydration synthesis is basically when you take water out of a covalent bond between 2 monosaccharides. So in this example, we have 2 basic amino acids, and we are taking one H from one of them, and then the HO, and that will give us H2O, because remember we have 2 H's, and then an O, making one water molecule. And then, so that is dehydration synthesis. But then when we talk about hydrolysis, we talk about adding water. So when you add that H back and add that HO back to this one, then they become separate, and the carbon is no longer bonded to the nitrogen, because that happens after dehydration synthesis. Sorry. But once you add that water back, they are able to become separate compounds again. So a way to remember hydrolysis is basically hydro, which means water, plus lysis means break apart, so break apart with water, and then dehydration synthesis. Basically, synthesis is the makeup of something, and dehydration, dehydro, take away water, like this. And so now let's talk about some examples of carbohydrates. They all include sugars and starches, and simple sugars, like we said, monosaccharides, are glucose, fructose, and galactose. Double sugars, which we'll talk about, are disaccharides. Maltose is basically malt, glucose plus glucose. Lactose is it's a very common sugar. It's what's found in milk and all those dairy products. Glucose and galactose. And then sucrose is table sugar, glucose and fructose. So as you can see, glucose is that main reoccurring sugar that we see that makes up most of these other, I guess you would call them subsugars, but yeah, glucose is a very important sugar. When we talk about a chain of disaccharides, we go into polysaccharides. I know we're getting a little more advanced, and we're progressing into a lot of sugars, but just stay with me. And this discusses starches, cellulose, and glycogen. These are just examples. And then the function of carbohydrates. So the overall function is basically to store energy. This is starch in plants and glycogen, animal starch in animals, and they serve to make up every cell wall in some organisms, which is cellulose. Remember, cellulose is the most abundant energy storer, energy processor on the planet, I guess you'd say, and it's very crucial to life. So now lipids. So lipids are basically your fats, your oils, your waxes, and we'll talk about that. But for now, we talk about how they contain carbon, hydrogen, and oxygen, like carbohydrates. But this time the ratio of hydrogen to oxygen is 3:1, and the ratio is not the same from lipid to lipid. So the hydrogen to oxygen ratio may differ. So now some lipids form from dehydration synthesis of three fatty acid molecules. Carbon chain, remember, with a carboxyl group at one end, and one molecule of glycerol, 3-carbon alcohol. So this is important. Triglyceride, 3 fatty acids, and a glycerol. There are 2 types of fatty acids: a saturated fatty acid, which contains no double bonds, and hydrocarbon chain. Carbons are saturated with the most hydrogens as possible, and unsaturated fatty acid. There are double bonds between some of the carbons in the hydrocarbon chain. So throughout time, a little sub-story, people thought at first saturated fat was bad for you. So then they came up with unsaturated fat. And then they realized that that was even worse for you. And so then it became all these food companies were trying to come up with ways to say, oh, this is healthy for you, even though it wasn't really healthy for you. And so this is like an ongoing debate that is an issue even today. So we'll see how this progresses as time goes on, but that's important, so don't forget that. So examples of lipids, like we said, fats, cholesterol, waxes, and oils. And the function of lipids is primarily used as sources of stored energy, secondary to carbohydrates. So carbohydrates will be the first, but then lipids will kind of be at the backup, and make up components of cellular structures such as the nuclear and plasma membranes. And they also cushion organs in animals. So like when we talk about our fat, this is kind of our lipids. It's what like kind of people try to get rid of, but when you really think of it, it's not terrible to have a lot of fat, because when it comes down to it, and if it ever came to starvation, just theorizing, your lipids would last longer, and they would go longer, and you would be able, when it came down to it, and you needed energy, your lipids would last longer and you'd get more energy. So now let's talk about proteins. Composition of proteins. So proteins contain carbon, hydrogen, oxygen, and nitrogen, and in some instances, however many instances, sulfur. So amino acids, they are the building blocks of proteins. Remember, we talked about them. This is the basic structure for an amino acid. It's non-functional, but like once you add more to it, it will have a purpose. And so amino acids, what are they? They contain an amino group, NH, negative NH2, a carboxyl group, negative COOH, and another group called an R group, a negative R group, which is a carbon-based side chain that varies in composition. The negative R group varies each amino acid in its special properties, or the negative R group gives each amino acid its special properties. About 20 basic amino acids are usually found in living systems. Some special amino acids are formed by modifying a basic amino acid. Like we said, this is the basic amino acid structure that's non-functional, but once you add more to it, it will do stuff. Alright. Stay with me. So now we have a peptide bond. A bond between carbon and nitrogen that forms between amino acids. So, when we talk about peptide bonds between amino acids, nitrogen and carbon, that is a peptide bond. And then amino acids are linked or bonded together with peptide bonds forming proteins. And proteins are formed when two or more amino acids are bonded together in an amino acid chain. So now dipeptide bonds, or dipeptides. A protein containing two or more acids in a chain. And then a polypeptide is a protein containing two or more amino acids in a chain. A dipeptide is a peptide with only two amino acids. Most proteins consist of hundreds or thousands of amino acids linked together in different combinations. Each combination being a different protein. So, examples of proteins: insulin, this regulates your sugar levels. Hemoglobin carries the oxygen in your blood. Enzymes, necessary for every cellular chemical reaction to occur. You cannot live without any of these. They are all crucial, but enzymes being the most crucial, as you would not be able to function at all without enzymes. So, structural protein. This is the components of cell structures and organelles, little organs inside cells, basically. And then function protein, enzymes that catalyze or speed up chemical reaction. So this is where we're going to stop, because we've gotten about halfway through the notes, and everything else from this is pretty forward and much faster than this. But if you have any questions, just go back in the video, and yeah, that's just half of it. So just stay tuned for part 2, I guess.